when I first saw the retina excised from the mouse under a microscope, lighting up in response to light, I was blown away. I was like, you don't even need the brain to do this. There's a lot of misconceptions about the retina acting like just a camera and then sending pixel by pixel information to the brain, which in some sense it does, but there's a lot of information within those pixels, and a lot of that information is actually being processed in the retina specifically. You don't actually need visual experience to get a robust direction selective response in the retina. We asked this question one step further. Okay, so what is leading or guiding the development of the circuit? And interestingly, before the mice open their eyes, the retina is spontaneously active, or the neurons in the retina are spontaneously active.
But if we really dive into it, how exactly is the retina able to detect motion? Why can your eyes detect motion before you're even aware of it? How does the brain turn simple light signals into something as complex as movement? And what does studying tiny circuits in the retina reveal about how you see and even how the brain develops? These questions point to something deeper. Vision isn't just about the eyes, it's about the neural circuits that are built, refined, and interpreted by the brain. Hello, I'm Avnish, and this is The Early Perspective. Each episode, we explore how the brain works and what that means for how we learn, think, and live. Today's episode is about the journey into neuroscience, what it's like in the field, and how something as specific as motion in the retina can reveal about the fundamental principles of brain development and function. To help guide us through this entire journey, I'm joined by Karina Bistrong. She earned her PhD in neuroscience from UC Berkeley and did her undergrad in behavioral neuroscience from Northeastern University. Across her training, she's explored how the brain develops and processes information, from her early work studying neuronal populations and the blood-brain barrier, to systems neuroscience research on somatosensation, and later her PhD work on visual circuits and how they develop and support motion detection. What's especially fascinating about her work is how it connects the different levels of neuroscience, from individual cells and circuits to ultimately how we perceive the world. She's now a postdoctoral researcher, continuing to expand her approach to studying how the brain integrates sensory information and also controls movement. Karina, thank you so much for being here. I'm really excited to talk to you today.
Awesome. Thanks so much for the introduction, for the invitation. I'm really excited as well.
Great. Okay, perfect. So let's just start with your journey. I know neuroscience is, of course, a very broad field, and it ranges from behavior, molecular biology, and even computational models. So how did you start getting interested in neuroscience, and what led you to specifically studying vision and the retina?
Yeah, that's a great question. I mean, I think when you typically learn about neuroscience, it's not until, you're already an undergraduate or maybe through some specialized programs later in high school. So when I was a high schooler, or even when I was younger in schooling, I actually did not know what neuroscience was a subject, but I always gravitated towards science, and I knew that I wanted to be a scientist, even though I wasn't necessarily sure what that journey would look like. And so when I was applying to my undergraduate institutions, I was applying as a botanist major or a botany major and a biology major. Because I was really interested in cells and molecular neuroscience, particularly in the natural world. And it wasn't till my undergraduate training that I switched my major to behavioral neuroscience, because where I went to college, Northeastern, had this really awesome program where you get to work full time for 6 months out of every school year, every other semester, pretty much until you graduate. And you can do up to 3 of these, and most people do 2 of them. And they're called co-ops. And when I first took my first biology classes and some electives that were in psychology, I found out that neuroscience was actually this great bridge between both, where you had the understanding of the human mind, but also this integrated my love for cellular biology. And so I got my first opportunities to work hands-on in labs at Harvard during this time in my undergraduate, that really kind of fortified my interest in not only just research, but also neuroscience specifically, because like you said earlier, it really does bridge the gap across all of these different scientific fields.
And I heard that you mentioned botany. That's really unique, because I've not heard anybody who's gone into majoring in botany. What made you choose botany first of all?
Yeah, that's a great question. I grew up in San Diego during most of my childhood, but I'm originally from Miami, Florida. And my dad in Miami is like a landscaper, landscape designer. He makes these really intricate and like grand waterfall landscapes for people in their backyards or for
Hmm.
different event spaces. And I just grew up, whenever I'd visit Miami, I grew up around a lot of trees and a lot of plants, and I was always really interested, kind of in just like how the fundamentals of the natural world really like kind of allowed evolution. I don't know if that's like that totally makes sense, but I always just had
Yeah,
like a
it
keen
does.
interest in that. And then during my high school classes, like I loved AP biology, but like my favorite sections were always like photosynthesis, learning like the reproduction of the plants, like all the parts of the flower. I feel like I was that weird kid that would like walk around with my friends and like name all the parts of the plant. Um, and so I felt like, if I couldn't get into any biology programs, I also wanted to apply to some botany programs. And there was really cool programs like up in Northern California that like specialize in this. But I'm glad I stuck with, biology because I don't know if I would have been able to go from plants to the brain as easily.
Then, once you did pivot to neuroscience, how did you find vision particularly as something you were really interested in pursuing?
Yeah. Well, I guess the short answer is, which I think maybe throughout this conversation you'll kind of get a sense of, is I have really broad interests in terms of like scientific interests and scientific questions. And when I was going through, these research opportunities that I had in college, I actually first started out in undergrad at Northeastern in like a clinical psychology lab. And that's kind of when, after that experience, I knew I didn't necessarily want to do like cognitive or human neuroscience. And then I got this opportunity at Bernardo Sabatini's lab at Harvard, where it was really my first like hands-on in the lab, experience. And we studied the basal ganglia and different cell types in the cortex that are in communication with the basal ganglia and circuits that are important for learning. And then down the hall, another PI was looking for a person from Northeastern to kind of do their rodent surgeries or have who had experience doing these types of techniques. And because of my experience in Bernardo's lab, I did. And so I got called down the hall, which is the lab that studied the blood-brain barrier. So I kind of just like followed that route, just naturally like went and studied that really piqued like my cellular biology interest because we studied the brain, but in this really molecular way, but also integrating still neuronal systems. Like it was my first time ever seeing neurons respond to stimulation in the brain, but the blood vessels also respond to that stimulation, which was just like really amazing. And then I ended up going to Fascination, and then I got into my PhD programs. And I think what's really great about a PhD program is, or going into a PhD program with this open mind, is that you never really know like which labs are taking students or how like the lab's research interests change over time. And so I went into my rotations with a really open mind, and I actually had not really known much about the retina. I didn't know that was like a field in neuroscience that people really like I was just it was so different from what I've been used to in the lab setting because we're not studying like the whole brain anymore. We're looking at just one part of it, but what's actually outside of the brain and in the eye. And I felt like when I first saw the retina excised from the mouse under a microscope, lighting up when you shot like lighting up in response to light, I was like blown away. I was like, you don't even need the brain to do this. And the fact that it can still do that without any extra processing from the central nervous system, or I guess that's like a contended idea, but from the central brain area.
Yeah.
I thought that was super cool. And then I guess in my interest that really like I think I was able to tap into by studying the retina are how neurons, learn either through development or through experience to form these really specific connections that really allow a system to function in order to process information both effectively, efficiently, um, and yeah, I think that's like why I chose the retina. And then in the time during my rotations, I really felt like I was being pushed out of my comfort zone, and I just felt like it was the right place for me to continue my training for sure.
Hmm. So it sounds like a lot of your motivations have been from finding something exciting and wonderful at a lab, and then you really leaning more into that, right?
Yeah, definitely.
Is there anything specific that you found at your current research that you want to lean more into, which is not just vision?
Yeah, that's kind of why I chose. So now I'm studying the motor system in
Mm-hmm.
mice, particularly the circuits that mediate forelimb movement. And this is actually something that's been, studied for a long time in many animal models because there's a lot of relevant reaching behaviors in humans and a
Mm-hmm.
lot of movement disorders that impinge on our ability to execute motor behaviors like walking or using our limbs for any sort of interaction with our environment. And I was really, thinking for a long time like what I wanted my postdoc to be in, if I wanted to continue doing visual neuroscience, if I wanted to totally pivot, because it seemed like that's just was has been kind of my vibe this whole time, was trying to
Mm-hmm.
be excited about different things and try to bridge the gap. But I wanted to be strategic because when I'm on the trajectory to hopefully, have my own research lab and become an academic faculty, and your work should reflect everything that you have done and also like tie it all together. And I felt like the motor system was a really great system to pursue because it lends itself to not only like movement, which is something I'm like, I was studying from the other side, like how we perceive movement, but now I'm studying how we
Mm-hmm.
generate movement, but also how you're integrating all of this sensory information through this like really distributed network across your entire body in order to execute rapid, fast, um, and either like learned or repetitive, reflexive behaviors. And so that's kind of why I shifted into motor circuits, but it still lends itself really to all of my other previous interests, which I think
Mm-hmm.
I'm really excited about. I've only been in my postdoc for about 3 months now, um, and so far it's been really rewarding.
That's great to hear. I think your journey is very wonderful in the way that your interests have kept on building upon the previous one and essentially kept kept moving, yeah, throughout your journey. Thank you for sharing that. I think
No,
that
of
was very important
course.
for people to hear. And then since we're on the topic of vision and the retina, I think it's important to first establish the foundations about that. Of course, vision is something that happens in the brain and a lot of processing starts a lot earlier, but what exactly is the retina doing beyond just detecting light?
Yeah, that's a great question. I think a lot of, there's a lot of misconceptions about like the retina acting like just a camera and then sending like pixel by pixel information to the brain, which in like some sense it does, but there's a lot of information within those pixels, and a lot of that information is actually being processed in the retina specifically. And what's really like, I mean, the retina is just like an amazing tool to study, representational view, like representational readouts of neurons, because they genuinely do represent like different neurons in the retina represent different features of our visual scene. And so the neurons that actually are just the light detectors are photoreceptors, aside from a few other retinal ganglion cell types that are light sensitive intrinsically, so without actually being photoreceptors, um, based on like genes that they express, but that's like a different topic. But once the retinal ganglion cells, which are the output neurons of the retina that send the information about the visual world to the brain, by the time they get that light information, there's a lot of pre-processing that's already happened. And so different retinal ganglion cells and even different interneurons called amacrine cells, and also the bipolar cells that are the intermediate excitatory cell type, they all care about different features within the environment. So some cells like only care when the light is on, some cells care when the luminance changes to dark, which also lends itself to being selective for different contrasts and edges. We have a lot of edge selected cells, which is really just kind of, where the border between contrast is. And then there's also motion sensitive cells. Um, and the way that these circuits kind of, or these cell types kind of accomplish this selectivity is really by the intricate or specific connections they form within the retina, within only a few other cell types. And so that allows that allows a lot of complex computations to happen within just a few types of cells, or not a few types. There's around like 45 or 60 something 45 retinal
Mm-hmm.
ganglion cells. Um, and so by having these really specific connections between the cell types, you're kind of maximizing the amount of features that you can really extract at this first level of vision.
So of course there's a lot of processing, a lot of steps, and a lot of complex cells which are involved in this process. But then, what do you think is a very simplified explanation of exactly what's happening every time your retina detects light or sees things?
Yeah, I mean, it's a really complex, I mean, that's a hard question to answer so simply. But if I had to use, like the motion detection circuit, like for example, we could, there's two, there's two types of motion that your retina is processing. There's motion induced by yourself moving from the environment. So you're experiencing, optic flow, or like when you're on a train and like the world's moving by fast. Um, that's called global motion, and then there's local motion, which is an object moving um, within your visual scene. So in the circuits that I studied the local motion detecting circuits, and you can really break down the computation of this circuit to two cell types. So one is the direction selective ganglion cell that cares about one direction of motion, which means if motion is moving to the left, it fires an action potential. If it's moving to the right, it does not. And then the starburst amacrine cell, which is an inner neuron in the retina, that has a circular morphology, and these neurons prefer motion moving outward from the center of their cell body out to the ends of their dendrites. And the way that leftward motion is accomplished is by having these neurons form really specific connections with, let's say, the right side of this circular neuron that happens to be inhibitory. And so now when motion is moving to the left, you're going to get no inhibition because that circular cell is not activated. But when motion is moving to the right, that circular cell is activated along its right dendrites, which releases a lot of inhibition onto this leftward preferring neuron that then shunts its firing. And I know that sounds really complicated. Like I'm trying to make it simple, but I really was like blown away when I first saw these, this connectivity and the inputs onto these neurons, how different they are depending on the direction of motion, and it really does boil
Hmm.
down to just the specific connectivity between these two cell types.
So different uh, directions of motions can affect the kind of connectivity between the neurons and the kind of signals being passed.
Yeah, it's more of the different connectivity between the neurons allows for the
Hmm.
detection of different directions of motion.
Oh, right. Okay.
And that's the type of that's the connectivity. We call it it's called asymmetric connectivity, where you have greater number of synapses for one on one part of this circular dendrite versus the other part of the circular dendrite of that same cell type. That's going to allow you to receive different levels of inhibition depending on what direction you're perceiving.
Right. So then, especially with the amount of neurons and a lot of processing involved, is it fair to think of the retina as almost an extension of the brain?
I mean, yeah, you could think of, there's a lot of, I think what's really nice about studying the retina that I now, now I'm studying behavior. I don't record from neurons
Hmm.
at the moment, is that you have a lot of access to study these microcircuits. And there's a
Hmm.
lot of, um, I would say, circuit motifs that exist within the retina that also exist within other areas of the brain to allow for these complex computations to happen. Like, for example, inhibition shapes and gates a lot of information in in the brain, and this has like been well studied in a lot of different systems. But the ability of this one cell to prefer motion along its radial morphology is actually due to dendritic properties that these neurons have. And these are like fundamental properties of neurons that allow, their dendrites to facilitate the electrical signal or facilitate the electrical signals that they need to in an effective way. Um, and like that's been seen like in hippocampal neurons, or these are really been studied in pyramidal cells in the cortex. And so these like fundamental principles that guide like complex computations exist within the brain as well, I would say. But an
Hmm.
extension of the brain, I'm not so sure, but definitely just as complicated and complex. Like
Right.
it took me 6 years to study and understand how
Uh-huh.
two neurons can, really, ultimately perform this computation. Of course, they receive inputs from the other parts of the retina.
And of course, that's also
Yeah.
the tip of the iceberg, right?
Yeah.
There's a lot more than that.
Yeah. I mean, yeah, there's, and I think the other part that was really, exciting and interesting about moving into the retina, is because it's such an evolutionarily conserved structure.
Hmm.
Most, vertebrate are visual creatures. And the like evolution of the eye, I think, is like one of the coolest and interesting, ways that we've been able to do so much with so few cell types.
Right.
And then that has like been sufficient to drive our complex behavior all throughout evolution.
Yeah, that makes sense. And then, so it does the brain process things like motion early on in the early stages rather than the later stages?
Yes. So there are different areas of the brain that get information about this global motion versus this local motion. And the retina sends, or these direction selective ganglion cells send their inputs to the lateral geniculate nucleus of the thalamus and the superior colliculus, and then they also get routed into visual cortex. And then the global detecting circuits or neurons send information to deeper brain regions that are important for like gaze stabilization, ocular motor function. And so the direction selective neurons that send information about local motion right to the thalamus and the colliculus actually have been thought to just inherit the neurons that get this information are thought to just inherit their direction and selectivity from the retina. And then it's been, kind of debated actually in the field. And I don't know if there's been any more recent work on this, so I could be not up to date. But there's been work showing that, these direction computations might also happen de novo in these brain regions where like local processing can actually make a direction selective cell in the cortex, for example. But if you look at neurons in the nucleus, they've shown that depending on the types of inputs that these cells receive from the retina, they can conserve the directionality of those neurons in these brain regions before it gets sent to the cortex. But there's definitely more processing, that occurs to get, more structure within the scene and the percept in these higher brain regions.
Mm-hmm. And then to kind of pivot to your other research focus, that is, of course, motion detection. I think that was really fascinating because, of course, we experience motion constantly. That could be just a car passing or just us scrolling on our phones. But we almost ignore the fact that our brain is detecting it and how it is detecting it. It's something so natural that we're forgetting it. But if we really dive into it, how exactly is the retina able to detect motion?
Yeah, so I mean, these are great questions, and I mean, that's a good observation way to think about it is that we are, kind of visual information is kind of ubiquitous around us all the time. And so how do you know
Mm-hmm.
what to like really pay attention to versus not pay attention to? Um, and one of the parts of, one of the challenges that our brain and our nervous system has to accomplish is kind of like, there's not kind of, it's filtering out all of the redundancies or the um, non-informative information that is pertinent to the action or the motivation of what we're actually trying to do or what we're attending to. But in terms of how we're actually like detecting motion, it really, like I mentioned, comes down to this microcircuit really in the retina, like I was alluding to earlier. You can take the entire retina out of the mouse, keep it alive on a dish, and shine moving bars of light. This was the technique I did in my PhD work. And you can shine moving bars of light, and you'll have one neuron that only cares if the motion is moving to the left, and it will not fire as strongly for the motion moving to the other, any of the other 8 directions that I show it.
Mm-hmm.
And so this like fundamental detection of motion is already accomplished just by these retinal ganglion cells in the retina.
And then is that just generally those kinds of direction selective neurons, the circuit that is detecting motion, or is there more to that?
I mean, there's other, motion sensitive cells in the retina for sure. The direction selective ganglion cells are not the only motion sensitive cell in the retina. A lot of or a good amount of the cells in the retina also can be selected for multiple features. Some, the direction selective cells also care about when the light is on or off. But they kind
Mm-hmm.
of need that to be able to detect a moving edge, for
Mm-hmm.
example. And so that, their multi-feature selectivity allows them to really, um, perceive motion. But yeah, they are not that like we don't only see directions of motion, right? We see all kinds of
Yeah.
motion. So
Yes, of
yeah,
course.
it would be inaccurate for me to say those are the only, that's the only type.
Yeah.
But those were these were this was the circuit that I studied for my PhD.
Right.
And so that's just what I have my
Hmm.
expertise in.
So then, once they detect motion in a specific direction, for example, what happens? What's the next step in that pathway?
Once the retinal ganglion cells get their inputs
Hmm.
from the upstream neurons that are, a part of this circuit, yeah, they send their information um, like through the optic nerve and into the brain. So that this goes into, like I mentioned before, the thalamic regions, and then
Right.
the spirit, and into the spirit colliculus, and then into the cortex. Um, and then, of course, depending on the type of motion information, this gets routed to other regions for, sensory integration. If you're getting information about, a visual stimulus that needs to, enhance your ability to run away from a threat, that information is getting sent to all the other regions of the brain that are going to integrate that and kind of, um, execute the appropriate behavior in response to what you're saying.
So essentially, once it receives the information, it kind of passes it along until the brain it is able to execute it and send it to different parts of the body, right?
Yeah.
Okay.
Or yeah, they would talk to other systems in the brain and
Right, yeah,
ask
yeah.
questions like, is this a threatening stimulus? And there's some features of stimulus that like are inherently threatening, like to a mouse on the ground, for example. All it really needs to see to feel threatened is like a shadow over it, because most of the time that's a bird coming down as prey. Um, and and in the labs, people have studied this stimulus actually quite strongly. It's called a looming stimulus, but in the lab, they'll, instead of obviously showing a bird over the mouse, they'll just have an expanding dark spot projected above it, and
Hmm.
it will um, engage in either escape or freezing behavior. And the circuits in the retina that are important for this type of visual stimulus have been extensively studied as well. But you can imagine if you have to execute a either an escaping or a freezing behavior to like save your life, other regions of the brain are going to be important in getting that information as well.
And then how's the brain able to differentiate between these types of visual stimuli? Like how does it know if something's is threatening versus something's completely normal and fine?
Yeah. I mean, I feel like it's like in the mouse, I feel like we show them quite, degraded stimuli, right? It's just, high contrast moving dots or bars. And that's
Hmm.
not the natural stimuli that like we all experience in the world, or that mice even experience
Right.
in the world. And that's been a really big question in visual neuroscience is, so how do the statistics of a natural scene actually get processed and integrated by the retina downstream regions? And I'm not quite sure if I have like a perfect answer for you. Um, but what I will say is that in terms of if the stimulus is threatened or not, you need to have integration of other sensory modalities and like other aspects of your nervous system are also
Right.
responding to any these stimulus. It's not just all visually driven.
Hmm.
But what the features of your visual scene that could be sending, like that is sending these signals, at the very big, occur at the very first stage, which is at the retina, because you do have or I mean, okay, at least in the mouse you do have these like really uh representational view already within in the retinal ganglion cells themselves.
So do these circuits change over time? For example, does a visual experience, like actually seeing types of motion, play a role in shaping these circuits?
Yeah, this is a great question. And this was essentially the basis of my thesis work was, um, so I guess, yeah, this was what kind of started my thesis project. I started joining a postdoc when I was a graduate student in the lab who was essentially asking this question: was does direction selectivity, or, the detection of direction of motion, require
Hmm.
visual experience? And what he did to test that was mice don't open their eyes until about 14 days after birth.
Hmm.
um, and then they opened their eyes. So what he did was actually keep them in complete darkness. So they were dark reared, and then probed these direction selective responses, like I had described earlier, by putting them in a dish and shining a moving bar of light. We're able to use fluorescent indicators to look at neural activity this way, and he found that direction selectivity was still there. And so you don't actually need visual experience per se to get to get like a robust direction selective response in the retina. And so then we asked, this question one step further. Okay, so what is like leading or guiding the development of this circuit? And interestingly, before the retina or before the mice open their eyes, the retina is spontaneously active, or the neurons in the retina are spontaneously active. Um, and this is what is known as retinal waves. And if you're looking at this activity, ex vivo in an explanted retina, and you have like a calcium indicator where you can see the depolarizations of the cells using a camera, you can see these like wave depolarizations across the entire surface of the retina. And so this is a correlated, structured activity across all of these neurons. And so we were then asking, could this type of activity be what's instructing or influencing the development of this circuit? And so what we did was manipulate these waves early in development by getting rid of an ion channel that really are required for the propagation of this activity. And when we did that, we found that not every direction was impacted by this manipulation. However, it was really interesting in that only the neurons that respond to horizontal motion, so motion moving left or right,
Mm-hmm.
are gone, or there was a sphere deficit, while vertical direction selective cells, so up or down, were still direction selective. And so we showed with that study that this spontaneous activity early in development before the eyes open is really important for detecting motion, but only along the horizontal axis. And that's like a really, that was like super surprising to us, because there's so
Right.
many hypotheses as to like what could be happening. And, if I was able to do another PhD, I would have tried to figure out what was going on with the vertical cells.
Mm-hmm.
To completion, this was also a part of my study. What has been shown in the field, or what kind of one of the hypotheses was that horizontal motion is actually the dominant motion that you get from optic flow. So when you're walking through the environment, that movement that you're seeing across your retina, or that's being projected across your retina, is mainly along the horizontal axis. And this is why it was really surprising that you don't need visual experience. And so maybe we thought that these waves, or this spontaneous activity, was kind of setting up the circuits to be ready to experience optic flow as soon as the animals open their eyes, because that's really
Mm-hmm.
important for mice, to navigate their environment. And we didn't find the molecular mechanism of exactly like why this is happening on one axis versus the other.
Mm-hmm.
But what I did show is that without that activity, those specific connections I was mentioning earlier between those two really important cell types for the circuit, is extremely disrupted, or severely disrupted, I guess.
Right.
And so that activity is somehow, modulating the synaptic connectivity in this very
Mm-hmm.
specific way. And, that is, something I hope someone in my PhD lab will pick up as a rotation project, or maybe a PhD project, and continue
Mm-hmm.
trying to understand exactly what's going on there. Um, but yeah, so that is kind of the development of the direction selective circuit, but it's different in different animal systems. So there's a fish model that people study called the zebrafish, and they have, these waves as well. They have direction selectivity, but they
Mm-hmm.
actually need to experience optic flow and swim through the water in order for their direction selectivity to properly develop. And so different species kind of have different strategies to kind of get these circuits to go on.
Could you extrapolate any of your findings to humans, for example? Or are they too different?
So there haven't been direction selective ganglion cells found in the human yet. There have been um, there have been in some direction selective types found in the non-human primate retina, and so they just and so it's hard to really make, these like synaptic connectivity types of questions, like whether the same principles are applied to, to human retinal circuits. I can't really speak to. However, one of the aspects of my study was that in addition to the neurons in the retina themselves not being able to detect motion along the horizontal axis. The mice were also not able to track motion visually that was moving left to right. So their eye movements were also disrupted, and this type of phenotype is analogous to a human congenital um, disruption called a congenital nystagmus, where there's
Hmm.
like rapid oscillations of the eye. And when you knock out that gene in the mouse, there's a homolog of that gene in the mouse. You actually do see this similar disruption of retinal direction selective ganglion cells along the horizontal axis. And so, while there might not be exact, um, the exact cell types that exist that we haven't found them yet in the human, there are implications for needing motion detection to happen
Right.
effectively and efficiently for many different functions to occur in the brain.
Hmm. Okay, so I know we talked a lot about your PhD research, but what
Hmm.
exactly are you doing currently? Like, what's your day-to-day research looking like?
Yeah. Well, I actually just got, like I said, I was only in my postdoc lab for 3 months so far. I just got my first set
Hmm.
of data last week.
Hmm.
And I'm in the process right now of applying to two fellowships.
Hmm.
And so, in lieu of that, I've been, yeah, trying to get some preliminary data for my proposals. But yeah, right now I'm going into the lab and I'm running some animal behavior. I do a lot of animal surgeries, so different injecting different types of viruses into the brain to express opsins that we're interested in, or doing like anatomical mapping. So you can inject like a retrograde tracer into one region, and then see what other regions are connected to it by this, fluorescent tracer traveling backwards. Um, but what's really special about, or I guess different and really special about my new lab is that my lab's only a year old. I'm the first postdoc in our lab, and we have one graduate student and two RAs. And so my day to day actually looks a little different than my day to day during my PhD because um, we spend a lot of time like all working together. Or I work really, we spend a lot of time like setting things up and kind of getting things piloted. I didn't walk into the lab per se with like my own behavior rig like, already built. Like I had to build this. So I work really closely with my PI to get these things going. And it's really nice because it feels like we're all, learning together, which I really appreciate because I'm switching fields again. Um, so that's been really fun. But yeah, I usually like walk into the lab. I'm at NYU. I'm on the 13th floor. There's a really nice view. So I walk in, I have my coffee, and then typically like plan out my day. And then I'll either do an animal surgery, and then depending on if I have mice that are ready, because now I'm doing animal behavior. I'm not working in development anymore. We have to wait like 11 weeks for our mice to be old enough to do experiments on them, which is very different than the timelines I'm used to working on. And then we also do these injections, and depending on what virus you're injecting, I have to wait 3 weeks for it to express. And so I try to plan my experiments very efficiently so that I always am working on something and I'm not always waiting 3 week stretches um, for mice to be ready. And then,
Yeah.
I did two surgeries today where I injected, an opsin or a light activated, you know, an opsin with a light activated that will make these cells light activated. And then I implanted optical fibers in the brain regions that I'm interested in to eventually, activate or inhibit those regions during behavior and see how the animal's behavior changes.
Hmm.
But a lot of, rodent surgeries, tissue processing, imaging of the tissue, making sure that my injections are in the right places. But yeah, various back to my systems neuroscience beginnings for sure.
Yeah.
With these techniques.
I'm sure it's really exciting, especially being in a new lab and really discovering a new part of the field again, right?
Yeah. We go to a lot of seminars. We have a seminar every week in our department. And now that I've been, thinking about motor control and particularly motor learning, now every talk I go to, I'm like, it has some aspect of like sensory motor integration in it. And I feel like I didn't appreciate before, how
Yeah,
how ubiquitous like movement is for every
right.
type of behavioral readout, especially within like systems neuroscience and the implications it has, for um, modeling and robotics and disease. Like it feels like I show up to work every day like ready to learn something new, which is always fun.
What do you think is the most exciting thing about this future of the field in general?
Ooh, that's a good question. I mean, hopefully by the time I have to kind of embark on my own journey, the field, I'm sure it'll be in a totally different place with how quickly,
Yeah.
things kind of move, especially in the context of, yeah, like modeling and, understanding
hmm
brain function in the context of like neural dynamics instead of like single neuron activity. You're really looking at all these populations of neurons and how they're acting across the brain. I think that there's a lot of really exciting advancements being made to really try to understand like what the activity actually means and how it's impacting our behavior. But yeah, I think for me the most exciting part is going to kind of see like I'm writing a proposal right now and I'm not sure, hopefully in 3 years I'd have completed the project or 4 years I've completed the project I set out. But I'm really excited to see how it's going to change because these things like change really quickly depending on like what the data shows you. And new directions um, kind of kind of show up all the time and it's just like deciding about which direction to take. And so I'm kind of I think it's interesting that all out of all of the choices I've made to throughout my neuroscience journey, I somehow ended up
hmm
back in the basal ganglia and the original, my first experience ever in neuroscience. And it's a huge field and I'm very intimidated by it, but I'm excited to kind of yeah, jump right back in and just kind of see where it takes me after this.
Yeah, it's almost a circle of life coming back
Yeah,
to where
exactly.
you started.
Yeah.
Okay, last thing now. If somebody's listening and wants to take away just one thing from your work, not just about vision, but as a neuroscience as a whole, what would you want them to remember?
Ooh. I mean, I think that the most important thing, or I think for me, is or like that I really gained from like my own experience that I think people kind of, when you're an undergraduate and I was pre-med originally, you're really focused on, you think you need to have this like one question, this like one interest, and you have to have all these things figured out in order to move on to the next step. And I would really like urge, anyone to just like, keep asking a lot of different questions. There's no questions that are, too, like there's no such thing as a stupid question. And really just follow your interests and try to be more flexible and spontaneous with what is really exciting to you. Because if you, approach science through the lens that everything is a learning opportunity and there's a lot of really good questions out there to ask and a lot of really exciting work to be done. You can always kind of make it whatever you
hmm
want to be as long as um, you know, stay motivated and put yourself, out there to keep asking good questions. But I would say, yeah, just be fluid, be spontaneous. You never know what the biology is going to tell you.
Yeah, that's wonderful, wonderful advice. Karina, thank you so much for breaking down something as seemingly simple as motion detection, but that which ended up really being complicated, but you made it really easy to understand. Thank you.
Thanks, I appreciate it.
And from exploring different paths in neuroscience to understanding how the retina is processing motion, this conversation really went to show how studying small systems can really reveal big principles about the brain. For listeners, if this episode helped you see neuroscience in a new way, or just made you curious about how your brain processes the world, Share it with somebody you think would find it interesting. If you guys enjoyed this episode and learned something new, make sure to follow on your favorite podcasting platform or on YouTube. I'm Avanish, this is The Early Perspective, and I'll see you in the next episode.